Hello and welcome to the Practical Animal channel with me, John Beaumont. Today we talk with Professor Tom Gilbert of the Centre for Evolutionary Phylogenomics at the University of Copenhagen's Globe Institute. Tom, welcome to the Practical Animal channel.
Thanks, John.
No problem. Tom, I read some of the research carried out by the Centre for Evolutionary Phylogenomics, and a whole new science was revealed to me. I mean, wow, what can I say? It's like an entirely different vocabulary. I had my veterinary dictionary by me, and I've been looking at it throughout the day. It's never been consulted so much. But what I want to do is get straight into the interview. So I will start by asking, Tom, who are you?
Yeah, well, so my name's Tom Gilbert. I'm a professor at the University of Copenhagen. I'm a biologist by training, but to be honest, I'm not a very good biologist. I studied biological sciences at Oxford, but I kind of actually fell into it by default. And I'm one of these, I'm a terrible organismal biologist. I really struggle to, recognize repeatedly the same organism. So you send me out to do field work, I just, I struggle. But I basically ended up being a geneticist, essentially, in studying DNA, and an evolutionary geneticist, so really studying evolution and ecology using genetic tools. And actually through that I moved into what's called genomics, which is just sequencing more and more DNA. So we now study organisms at the genome level. And actually most recently I've transitioned into this term you said, a hologenomicist, which is a kind of new area. I think a lot of people still kind of view it as some kind of like made up pseudoscience, to be honest, because it's a bit different. But essentially what our interests are is studying how life works, but not just from the point of view of the organism, not just the point of view of the big thing, the plant or the animal, but also the microbes. What are the microbes doing? And how, if you really want to understand something basic, such as how does a cow eat grass, or how does a certain kind of bird eat a certain kind of thing, or how does a vampire bat drink blood, you really have to understand it as the interaction of both the animal and its microbial passengers. You view it as the whole thing, and that's really what holo is. The hologenome is studying the genomics of both of them, both the union of the organism and its microbes. And that's really what we do.
I'm going to go off script slightly here. One of the first things that hit me when I was looking at the website, the research that you do, was a couple of basic terms that I first came across 25 years ago, more than that, and they just completely slipped my mind what they were. So can you very briefly explain what is a eukaryotic and a prokaryotic cell, please?
Sure, yeah. So yeah, these are old terms. I mean, essentially, life is split into several major domains, and we have the eukaryotes and the prokaryotes. And the prokaryotes you'll basically know as bacteria. It's tiny microscopic organisms with very small genomes, some of the most ancient things on Earth, and most of the pathogens, for example, people hear about every day are bacteria, prokaryotes. And then you've also got the archaea, which are also very like the bacteria, but are kind of a whole spin-off. The eukaryotes are basically they're typically larger cells. They're made out of more complex DNA, and actually it's been sort of argued and pretty conclusively demonstrated that eukaryotes actually evolved, weirdly enough, by the enslavement of the prokaryotes. So part of a eukaryotic cell is actually bacteria that were captured into it in the first place. They're almost composites to some degree, and one could even argue they're the first actually what we call holobionts, the sort of union of multiple individuals together. Just as how, for example, a holobiont is like a coral. A coral is an organism with a lot of bacteria that are part of it, but also another organism that's there. Actually, that's what happened when the eukaryotes were formed. We had these sort of some kind of microbe organism encapsulate another microbe organism, formed a union of the two. But to practical purposes, to be honest, eukaryotes are basically everything bigger than the bacteria. Whether it is an amoeba, for example, which is still single cell but larger, whether it's fungi, whether it's plants, whether it's algae, whether it's vertebrates, your animals, your mammals, and so on. It's all the stuff that's slightly more complex than the bacteria.
Is it fair to say that you are an evolutionary biologist?
I am an evolutionary biologist. I'm a much better evolutionary biologist than field biologist, put it that way. So I am, most of my research is anchored in understanding evolution.
Excellent. I'm glad that you mentioned the vampire bat, because we're going to come back to that in a couple of minutes. But can you tell me, what do you do?
What do I have a very glamorous job. I spend a lot of time doing admin, paperwork, bills and accounting. I mean, John,
Hello?
sorry, we
Yeah, I'm here.
broke up. So what do I do? I mean, I of course, like many people, for my sins, I do a lot of admin. But research-wise, what my colleagues and I do is we're trying to actually develop and propose new hypotheses for actually to explain some of the really big fundamental questions in ecology and evolution. So some of the questions that have fascinated biologists for a very long time, and including Darwin, going all the way back to there, is questions, for example, like why do some species survive for a very long time and even diversify into many, many, many other species, and other species either go extinct or they live for a very long time, like the coelacanths, but they don't diversify. There's very, very few of them. Actually, bats are a good example again there. Vampire bats, there's only 3 species of vampire bat, right? But there are hundreds of other species of insect-eating and fruit-eating bats. And a kind of fundamental question in biology is, why are there so few vampires? Why are there so many of the other things, right? Other fundamental questions that we're interested in is things like, what determines which animals did we domesticate? So why did we domesticate the wolf, but not the jackal, not the African hunting dog, right? Why did we domesticate cats, right, but not hyenas and so on? And even actually other bigger picture questions such as, why did the dinosaurs go extinct, right? Why did the mammals and birds survive and the dinosaurs go extinct? Now, these are big evolutionary questions, and people have studied them for a long time and they've got lots of answers for them. What we're trying to do is propose some alternate answers. It's actually interesting, I've actually talked about this before, and somebody said, well, you can't prove it anyway because it happened in the past. And that's completely true. We can never prove what happened in the past. We can come up with hypotheses of what might happen, and we're particularly interested in proposing new hypotheses that are maybe outside the normal understanding or outside the normal explanation. So I'll give you one very good example. If you talk to your average small child about dinosaurs, because small children know the most about dinosaurs, it turns out, why did the dinosaurs go extinct? The standard story is a meteor hit the world, right? And 66 million years ago, it caused a kind of nuclear winter. The dinosaurs died out, the birds and mammals survived. So then the question is, well, why did the birds and mammals survive the nuclear winter and the dinosaurs didn't? They were all there when the nuclear winter happened. And actually a kind of very standard textbook answer to that is, well, maybe the mammals and the birds were maybe nocturnal, omnivorous, a bit smaller. They could maybe have a wider range of things they could eat. Maybe they just could weather the storm better, right? Which is okay, but it ignores conveniently the fact there were dinosaurs in the nocturnal omnivorous niche as well, right? I mean, they were like, relatively cold climate dinosaurs. So that alone is not enough to explain it, but it's the standard argument. Now, we actually think there are maybe other explanations as to why, and we think that maybe the birds and the mammals, maybe they essentially had the capacity to diversify faster. They could sort of change their way they live, what they eat off, faster than the dinosaurs in this crazy new changing environment, right? And we actually think what allowed them to do that was how they could exploit the microbes inside them, because all your microbes do inside you is they're like little factories digesting the food, for example. And of course, if you have an ability to tolerate a very wide range of microbes, that allows you to actually eat a wide range of things. That gives you the ability to suddenly change your diet if you have to, because the nuclear winter's hit and everything's dying, right? So by us invoking this relationship with the microbes, it gives you an alternate explanation. And we would argue that the dinosaurs just simply were stuck with a much more narrow range of microbes and therefore couldn't do it.
Wow, that's absolutely fascinating.
I also hope you caught it, because
Would you consider yourself...
went up and down, but
No, I caught every word. Okay, so would you say that you are as well a conservation biologist, apart from being an evolutionary one?
Yeah, I would say I'm a bad conservation biologist, but that's because I also, to be honest, I have slightly heretical viewpoints on conservation. And so what I do is relevant for conservation. I don't necessarily ascribe to a lot of the dogma in conservation, which I guess fortunately I'm not vocal enough in the field, but I have some opinions that are maybe less popular. For example, genetic diversity, this is a very hot topic in conservation. There's this dogma based upon actually both lab experiments and theory that diversity is good. You need to maximize the genetic diversity. And, if you have low diversity, then your species is in big trouble, right? And so as conservationists, we should try to maximize diversity. And the take home of this is, for example, in East Africa, the Kenyan Wildlife Service are translocating rhinos to basically bring them together, and they're genetically testing them in the hope they can even breed them to sort of maximize the diversity, right? And the question is, though, to me, is there any proof that diversity is important, right? And there's lots of proof if you look at the extreme. I mean, the most famous one we talk about is the Habsburgs. The famous royal families of Europe were famously inbred, famously problematic, right? And if you also put, for example, Drosophila flies in test tubes for thousands of generations, they get horrifically inbred and problematic, right? But my question is, maybe how often does the wild get to that extreme, right? And are we actually maybe forcing diversity where it's not needed? And that may sound like a strange thing, but for example, I remember reading in the last couple of years during the translocation of rhinos in Kenya, an awful lot of them suffer a lot. They die, right? Because of the stress of the whole process. And sometimes what you're doing is maybe more stressful than what's going on. So typically where I've been actually working in the area of conservation is in addition to this hologenomics, actually my background before that's what we call ancient DNA, and is sequencing the genomes of organisms, of not just modern organisms, but historic ones from collections. So for example, in this regard, I've had a few projects on things, for example, like lions and black rhinos and white rhinos and koalas, where we've actually tried to go to museums, get samples from 100, 150 years ago, sequence the genomes of these things, measure the genetic diversity then you sequence the genomes today, measure the genetic diversity and see has it changed. If in the last 150 years you have a measurable loss of diversity, then you can say, okay, well, something's actually happening. We humans are a problem, right? The diversity is being lost. On the other hand, if you look 150 years ago, and there's basically the diversity was no different to today, actually it suggests long-term it's had low diversity, and it's probably completely irrelevant. It's a red herring, if you will, right? So I've been trying to advocate that we can use these nice, sad, unfortunate collections that exist of charismatic mammals that existed because we hunted all the poor things, to actually study how their population diversity was through time, and use that to maybe inform conservation. Is diversity important? When is it important? And maybe move away from this default knee-jerk reflex that we have to maximize diversity. And again, it may sound like a petty thing, but it's about money. Conservationists do not have a lot of money, right? I mean, there's always the question, it's like triage in a hospital. What do you put the money towards? And, doing genetic testing of rhinos in East Africa and translocating them is expensive work, right? Do you do that? Or do you spend the money on education to, work with the locals to increase protection, right? It's basically what it comes down to.
Yeah, it's very interesting that you say that, because 7 years before I was in Malaysia with you, I was in Mauritius working on the Mauritius kestrel, which is a famous case because there were 4 individuals in 1974, and there are now 280. I did a video on it last week, and there are now more than there's ever been historically.
Yeah.
And people were concerned about genetic diversity, but in actual fact, in the real world, it seems not to have had that much of a difference. Interesting.
Yeah, no, that's exactly right. Actually, you'll probably like this. I actually have a postdoc right now, a very nice Mexican man called Hernan, working on the kestrel down there. So he's doing exactly what I've said. He's actually going to be sequencing old samples and new ones to actually look at how the population diversity has changed, both before the bottleneck and after as a result. So for exactly that reason.
Fantastic. I've got a dozen questions I could ask, but I've got a few others which I've prepared, so I want to stick to those as well. So the Centre for Hologenomics vision is retelling the story of life. How does it do that?
Well, yeah, that gets back to what I was saying earlier. I mean, say we have an understanding of how life works. We're trying to advocate people should view life through this hologenomic window, which is, don't just look at the microbes, don't just look at the bigger organism, look at the union of the two, and use that to, sorry, John, I think I might have lost you.
No, I've got you. The signal is variable. I can hear you and I can see you.
Okay.
Your image keeps freezing, but I
I
can
don't know
hear
what
you.
it's going to do to your interview, but presumably you should be able to edit it or something to cut out the crap you don't need now.
Yeah, yeah, not a problem, not a problem.
Okay, but do you want to
The
ask
centre, yeah, sure. So it was the question was, which I'll repeat because the signal seemed to go. The Centre for Hologenomics vision is retelling the story of life. Tom, how does it do that?
Yeah, so it's a good question. I mean, essentially, we are tackling stories about how life happened, whether it's how the dinosaurs went extinct or why did we domesticate the dog, how do we domesticate the dog, why did certain things go extinct. But from a new angle, we're trying to propose alternate hypotheses to explain it. So we're trying to show that if you don't just focus on the dog in the domestication or the dinosaur itself, but focus on it and its microbes together, you get alternative explanations for what's going on. So that's really the retelling part of it. It's providing alternate hypotheses, alternate stories. So again, I'll give you another example that's dear to me that I think is a fascinating story. And I will clarify, this is just an idea. There's no proof yet. But, you know, there's enormous interest in dogs. I'm sure you know this. Everyone loves their dogs, right? And there's enormous interest in how did we domesticate the wolf to become the dog, right? A lot of people have studied this. Archaeologists have studied it. Geneticists have studied it. And it comes down to one real key issue. Wolves, you don't want to live alongside a wolf. Dogs, you can live alongside. We fundamentally changed the animal somehow, right? And the typical way to study it from a genetic point of view is you get a wolf and you sequence its genome. You get a dog and you sequence its genome. You look for the differences between them. You identify genetic mutations that do something. For example, you might find, in fact, for example, they've looked at the amylase gene in dogs, and dogs have got multiple copies of amylase genes, which basically means they can eat starch, right? Wolves aren't naturally predisposed to starch. Dogs are predisposed to starch because they have this mutation, right? So you look at the end product. And that's all good, but I'm actually interested in the start. So I'm interested in the question, how did we get through the first number of generations where we had this aggressive wolf-like thing? We managed to live alongside it and change it so that it ended up at the end point, right? And again, to put it into context, it's kind of like, if you want to look at the evolution of the car, right? You can look at what a horse-drawn cart looks like. You can look at like a very, very fancy Audi looks like today, right? You can see the end product. That doesn't tell you what happened in those first steps. When you get back at those amazing pictures of late 19th century early cars, that tells you the early steps, right? What we're trying to do is the same thing with regards to dog domestication. How did we get through the early stages? What were the earliest things we had to do to eventually end up with the final dog? And I honestly think the biggest challenge we had was just making the wolf a stable animal that you didn't have to fear would eat you in the night, right? I mean, you know, I would not lie down next to a wolf. I would not, you know, I'm quite happy lying down next to a dog. So if you think about it, it's the behavior, right? And we actually are trying to think about how can you explain a very rapid change? How can you take an aggressive animal and instantaneously or very quickly change it to a stable thing alongside you, right? Now, again, this may not sound difficult, but people have not really managed to tame a modern wolf. If people tamed modern wolves just like that, people would be selling them. You'd be able to buy them. Everyone would want one of these things, right? There are things like Czech wolf dogs, which are about 12% wolf left, right? But they had to back cross with Alsatians. Right? So just getting a wolf and making it not a wolf behaviorally is hard. And this is actually where the hologenome idea comes in. So bacteria, they are little chemical factories, right? When you know, all bacteria do is they take things and they make chemicals out of them, right? And that's actually how you industrially make drugs. You know, big drug companies, they have big tanks full of bacteria making drugs, right? And we know inside us that bacteria do things. There's a lot of interest now in actually what's called the gut-brain axis, which is how bacteria take things, create metabolites that affects your brain, and that they're linked right now to depression and anxiety and all these kind of disorders, right? So to me, that presents a really simple mechanism for domesticating a wolf. You know, you have an aggressive wolf, you somehow modify its microbes inside it so that they produce essentially drugs. They basically drug the poor thing, right? Change its behavior instantaneously. You've now got a passive animal living with you. You don't have to do anything to the wolf's genome, right? You'd have to select on it for changes to make it less aggressive. Initially, you just purely drug it. Now, that's not to say humans consciously drugged them, but if you now think about a plausible, how did we first live alongside wolf things, right? I would gamble on the fact that it's the fact that, you know, puppies are cute. There is no doubt that humans find baby animals cute. And there's no doubt this is not a very ancient human trait, because it's human brains are hardwired to that kind of baby look, to look after our own babies, right? So I would not be surprised if your hunter-gatherers in Europe or Asia 30,000 years ago, either kill a mother, stumble across the puppies. What do? They're like, oh, that's a cute little thing. Let's bring it back to the kids, right? Once you've brought back a puppy, it's very easy to expose it to microbes that are different to its normal thing. And then you basically, if it's the right kind of microbes, they can immediately start providing drugs and therefore basically drug the thing. And bingo, you've got your initial thing. So our explanation is the microbes may have been critical to the process, and that is how we domesticated it, right? It's not all the end things that happen later, it's that first thing. So a very long answer to your question.
Absolutely. No, that's absolutely fascinating.
And I should clarify, not a shred of proof for what I just said then, but we're trying to get some proof out of it.
That'll be in the next in 2 years.
Yeah.
The Centre for Hologenomics has various research groups. Can you tell us about those, Tom, please? Yeah,
so we essentially have 8 research groups. So I'm the director of it, but I have my 7 other colleagues. And we were brought together because we have a shared interest in this alternate new way of studying life. But it's also it's a matter of skills, right? So, when I was studying genetics, so as part of my bachelor degree, 20 years ago, when you study DNA, you study very short fragments of DNA. You would, if you wanted to study a genome, you would sequence it in little fragments of maybe 500-600 bases at a time. That's kind of like studying the Encyclopedia Britannica, like a sentence at a time, right? It was very slow and painful. Genomics, there's been a revolution in genomics where we can now generate large amounts of data very, very quickly. Right? You can sequence whole genomes at once. That's like getting a whole Encyclopedia Britannica at once, right? But actually, to make sense of that information is difficult. And that actually has a huge mathematical, computational part to understanding that. And so in my center, we have both geneticists like me. We have field biologists who actually go and do the actual exciting experiments. We do animal experiments, and I don't mean that in the nasty drug company way, but I mean more like take some animals and put them in pens and actually watch them with like fancy cameras, what they're doing and so on. And then we have computer scientists. So a couple of them are hardcore mathematicians and computer scientists whose role it is to help us stick these massive amounts of data together and try and mine out of it what's going on. And that's like, this is this machine learning revolution that's happening in a lot of places. Essentially, it's witchcraft. It's these black boxes of computational methods where you put in large amounts of data and things come out. And people are trying to do that to predict the stock market, for example, or predict the weather or so on. We're trying to do it to predict the effects of certain microbes and certain organisms and all the data we get to see what's going on to try and support our hypotheses. So essentially, at the center, myself and my co-PIs, it's fulfilling the different angles. So we have the proper animal behavior experts, we have the conservation guys, we have the hardcore molecular biologists, we have the computer guys, all together so we can work as a happy commune.
Brilliant, fantastic. So you are head of the Centre for Evolutionary Hologenomics.
Mm-hmm.
How did you get to where you are today?
Oh, yeah, that's a good question, John. And that is, I am a very bad role model, put it that way. So when I met you back in '97 in the jungles of Borneo, I had just come out of school. I had managed to get in to study biology at Oxford, mainly by default because I wasn't good enough to do maths or physics. And I kind of looked carefully, like, which one can I get in on? It's not because I wanted to be a biologist, which is probably explains why I was such a bad field biologist when I was out there working as a field biologist on this like gap year. So, it wasn't really my plan to end up doing biology in the long run. My plan was to go to Oxford, enjoy being there, and then go off and get a high-paying job in a bank or something. And to be honest, I spent most of my undergraduate degree working out how to get a job in the city, as does half the people at Oxford, right? I can't say I'm proud of this, but it's what everyone did in those days. And I was actually all set. I had a job lined up in a bank. And then actually a few weeks before the end of my undergraduate degree, a guy called Eddie Holmes literally cornered me in the corridor and was like, "Do you want a PhD, Tom?" And I was like, sure, why not? I literally jumped to it. And I know this sounds extremely ungrateful, John. I know, not least because you know, but I honestly wasn't planning on it. And I actually, I did like what Eddie did. He basically studied viral evolution. I did find it fascinating. And I was like, what's the harm in postponing by a few years, right? I quite like being a student and so on. So I literally fell into PhD. And but actually, while I was doing that, I started to actually realize, okay, I quite like this research. And it wasn't, I never liked the research before. It's just, I didn't like the uncertainty. You know, what you hear about academic careers is it's all short term, short term, short term. It's, you know, a few years, get a grant, get a grant. And of course, all students hate writing. So why would you want a job where you spend all your time writing to try and get more money, right? I mean, you know, this is what really put me off it originally. But I did the PhD, and towards the end of it, I was actually, I really quite like doing this. And I tried writing some grants. I didn't get very far, but again, I was in the elevator one day before I finished my PhD, and a guy came in, a Canadian guy called Mike Warby. He was like, "Hey Tom, what are you doing in the autumn?" And I'm like, "I'll be unemployed." He's like, "Do you want to come to Tucson?" And I was like, I'd never heard of Tucson. I had no idea what it was. And I said, "Let me go and ask my wife," or she's my girlfriend at the time. And so I went home and said, "Should we go to Tucson?" And she was like, "Where's Tucson?" And we literally couldn't even spell the damn place. Like, I had some vague notion it was somewhere in the US. And she was like, "Well, I've got no better plans." And so she was working in Didcot Parkway, which is not the most glamorous part of the world. And so I sort of said to Mike, "Sure, we'll come to Tucson." And I ended up going to the University of Arizona for 2 years purely because it was better than being unemployed. But again, I really found I liked the research. And I was actually working there on HIV, actually. I was working on the origin and evolution of HIV, which is very fascinating, the evolutionary angle of it. And then actually, when I was down there, again, I still didn't have a concrete plan, but my girlfriend, who was then my wife at that point, is a Dane. I didn't speak Danish. I was fed up of not being able to speak Danish because I couldn't understand the family whenever I was back in Denmark. Oh, so I thought, well, I better move myself to Denmark to learn the Danish. So I actually managed to get an EU grant, a Marie Curie fellowship, to move to Copenhagen, where I knew some biologists, mainly with the intention of learning Danish, again, not doing the biology. And again, that's why I'm such a bad biologist. But again, I got the grant to go there to do more work on HIV. And I actually just, I mean, it's not like quite like Denmark, but I actually just kept getting more and more. You get really hooked into the research, right? And so this thing that was initially a kind of means to an end suddenly became what I actually wanted to do. So I just wrote more grants and found I was quite good at writing grants, found I was quite good at research. And then eventually, you know, as you get high enough in the system, more opportunities come up. And so this center that I direct now was actually yet another grant scheme that came out, and I applied with the wacky ideas, and I wasn't really expecting it because they are quite wacky ideas, but to my massive surprise, the foundation funded it. And I would say I'm very glad because I believe in my ideas, but typically the other people don't typically believe in my ideas. So it was nice that somebody bought into it. And then we ended up here. But I say, so I am a shockingly bad role model because I'm a terrible biologist, a field biologist. I didn't want to be a biologist, but that was partly, I think, because I didn't really appreciate the beauty of biology, and I was just scared by the career path. I mean, as you know, it's just academia is a brutal, brutal thing. So I was thinking, why in the right mind would anybody want to put themselves in that situation where you have such insecurity for such a long way? And there's this famous academic pyramid. You know, I saw an amazingly helpful report from the Royal Society a few years ago about career progression in the UK for STEM graduates, right? Science, technology, engineering, medicine graduates. And it's something like, of the people getting PhDs in the UK, something like 55% immediately leave academia. And then by the time you get to a faculty position at a university, like a lecturer or something, it's 2.5% are left. And by the time it's professor, it's like less than 0.5% are left. There's 1 in 200 are ending up there. So, so are you still there? Yeah.
I'm still here. I'm listening.
So the point, so it's 0.5%. So I mean, once you see these kind of figures, and it's like, whoa. And to be honest, it's actually, the report's a very positive thing. It's like 199 out of 200 get these great other jobs elsewhere, and probably most of them are earning more than the poor UK academics, right? But of course, if you're in it for the academia, it's this crazy narrow triangle. So, you know, it's scary. And that, to be honest, I mean, I didn't know the numbers, but I knew that early on, right? And that's why I was like, there's no future in this for me, right? But hey, luckily it worked out, and I'm very glad it did. And it's not very fair, to be honest. It shouldn't be me, but hey, that's the way life works.
Very interesting. Tom, how would you describe your leadership style?
Yeah, that's a good question as well. You should probably ask people who work for me that question. You know, I to be honest, I kind of think that the best thing I can do, given my talents, is facilitate things for other people to do things. So I understand politics. I understand how to get money. I understand big picture questions. I'm terrible at small details. So what I like to do is to facilitate other people. So for example, I don't like having students or postdocs who are automatons who do what you're told. I think, to be honest, that it's far too easy to get a PhD these days. I think that there are too many PhD students that are essentially glorified lab techs, right? I mean, it sounds crazy, but so many PhD positions are projects conceived by somebody else where they come in and do the work, but it's not their project, right? When a project's your own project, it means a whole different thing to when you're working on someone else's project. And to me, a PhD student should be a proper independent researcher who should be the expert on the topic. It should never be the PI, right? And I try and facilitate that. So although we are very constrained by a grant system where you essentially have to write PhD projects into every bloody project you write, right? Because they want to train PhD students. I prefer a situation where I can get hold of money to provide a PhD studentship in a kind of general area and then find people with a good idea and try and make it around their ideas as best as possible, which is not straightforward because so much of the money is very constrained to what you said you were going to do that, find someone to do that, right? But I do believe that the best independent scientists are the ones who have their own ideas and can test them out. So what I try and do is literally facilitate that. And this center I have is really that. I mean, I'm the head of the center. I sit on a very large budget, but I give most of the money to the other PIs for them to kind of develop their thing because I think that's what's important. I mean, it's if you want to just generate data, go work for a company or just hire some research assistants, right? The PhD students and the postdocs are the people who should really have the power to do what they want to do. So if I can facilitate it by providing money and resources and that kind of thing, that's what I like doing, to be honest. It's and also, there's one of me, there is X of them, they can generate X times more results, right? And for me, the satisfaction is seeing the results, whether I'm right or wrong, or whether they're right or wrong. It's the satisfaction of seeing it, right? It's not so much and it was all my idea. That's kind of bullshit.
That's great. So just to continue with the interview, Tom, you have published a paper on dog evolution. Can you explain the link between canine transmissible venereal tumor and dog evolution?
Yeah, I can. That's one of those tough questions because although I was on that paper, it's not at all my study. That is a study led by Gregor Larson at Oxford, and Laurent France, who are very, very good guys. But it's a really interesting story. I mean, it's so actually I and Gregor and Laurent, we're interested in this question of where dogs come from and how old are dogs and where were they domesticated. And the problem is that ideally to do that, you would not only study modern dogs, but you would study archaeological dogs from the past, right? And basically, what technically you do is you try and find subfossil dog material from all over the world, and you sequence the DNA in it, and you try and find what is the most ancient looking like dog seen, and where is it found. But that's surprisingly difficult because samples don't preserve everywhere, especially in regions that might be interesting. And Gregor actually and his colleagues, while doing this, they got talking to this other group working on this transmissible tumor, which is a very weird thing. It's literally like a kind of nasty organism that jumps onto animals. It's a tumor that literally can horizontally transfer itself, right? It's kind of freaky. It's like a cancer that jumps onto you. And the guys sequencing this realized that the cancer has its own dog genome, right? The genome of the cancer, the transmissible tumor, is not the genome of the dog it's sitting on, right? It's this whole weirdly transmittable entity with its own genome that is completely distinct to the dog it's sitting on, but it's still a dog-like genome. And while they were analyzing it in light of a big data set of dog and wolf genomes that we have and my colleagues have, they realized that not only does the transmissible tumor have its own genome, but it's actually a very primitive looking like dog genome. It's almost like the earliest dog-like genome there exists today, not in modern dogs, but in this weird transmissible tumor, right? And it actually is extremely closely related to dogs that are found in the Americas, essentially, in Greenland, right? Which is an extremely early basal branch off in the dogs. We know that when dogs are domesticated, there's sort of a fundamental early split, and one of the early splits heads off essentially into the Americas, right, out of Asia or Europe, wherever it goes. And weirdly enough, this tumor is basically carrying that, right? So this tumor, which is now found today on dogs all over the world, essentially, right, with its own unique genome, is carrying a genome of one of the earliest dogs, which is just insane. It's completely insane. This thing's also been transmitting around for 10, 15 plus thousand years with its own genome, infecting the poor dogs, carrying its own freaky genome. And it's literally as if you wake up one morning and you've got a big tumor growing on your cheek and it's basically got something inside it. I mean, it hasn't got a brain, but it's a whole living separate organism, right? So, the actual evolutionary point of view is it actually tells us a bit about dog domestication, and that actually that there's been migration happening in dogs, probably from the Americas back to Asia and stuff, to bring this, and it must have originally arisen like 15,000 years ago and so on. So it weirdly enough tells us about things about how old dogs are, where they come from, but all based on this completely freaky, weird medical condition today. That's a crazy story.
This is brilliant material. Thanks, Tom.
Yeah, I mean, it's like stuff out of a sci-fi film, out of like the Twilight Zone or something. I mean, it's horrific to think about this biological entity that literally can transmit onto you and grow on you. I mean, but actually, it's similar to what happens in Tasmanian devils, for example. They have these transmissible tumors and so on. So.
And something similar happens with Tasmanian devils?
Yeah, the facial tumors that kill the poor buggers. I think that's also a transmissible horizontal gene flow. I mean, transmissible tumor. That's how they get it, because they fight and bite all the time, and this tumor then jumps onto them, right? So it's a nasty biological entity that is essentially Tasmanian devil, but transmits onto new ones and infects them, but not like it's a virus or something. Yeah.
I feel I could take this conversation in a million different directions. It's fascinating. But
and I'll get less and less accurate to say moving forward I know nothing about, but yeah.
You've done research on vampire bats.
Yeah.
How is that work progressing regarding spread of rabies?
Well, yeah, that's a good question. I actually don't work on rabies, but I know people who do. I mean, my interest in vampires was actually what got me into this whole topic of hologenomics to start with. I had a PhD student called Lysandra, who was a Mexican, who was basically a bit of a goth, and she was like, we were talking about projects, and she was like, I really want to work on the vampire bat and how it is. I was like, fine. I said, you get the vampire bats, we'll do it. So we were actually studying this phenomenon of obligate blood feeding, right? How is it the vampire bats, the three species of them, are able to solely live off blood, which is it's sufficiently weird that very few organisms do it, especially vertebrates, because it's actually a very bad substrate for food. It's got no vitamins in it. It's got too much salt in it. It's got too much iron in it, right? It's got pathogens in it, right? So, it's not the wise choice to live off, but they've evolved to just eat on it, right? And so we were studying the adaptations in the genome of the vampire and the microbes in the gut and how they were working together to do everything from, for example, provide the vitamins. It turns out the bacteria in the gut of the vampire take the blood and synthesize all the vitamins for it, right? Then there were also small predatory eukaryotic organisms so basically small single cellular things in the gut that were there to basically try and take out nasty pathogens that could be in the blood and so on. And so it explains how it's going on. With regards to rabies, I mean, what we do actually doesn't help you very much, because it shows that vampires are quite happy drinking blood. It goes to the added benefit of the knowledge. Sorry, it adds to the knowledge about bats are remarkable reservoirs for pathogens, right? Bats, this is the whole COVID problem. Everyone's blaming COVID on the bats, which is probably true. But I mean, bats, and to some degree birds, can carry a very wide range of pathogens, mainly because their immune systems don't go crazy and don't kill them for carrying them. Because typically the way you die of a pathogen is your immune system kills you for, in trying to get rid of the pathogen, it goes overkill and kills you. And, that's why bats are so good at carrying rabies or coronavirus, SARS, Ebola, and so on, is because their immune systems are pretty chilled out. We weren't studying that ourselves, but some of the guys I work with are studying this. But the unfortunate take home is that they're the perfect reservoir for these pathogens, because they just literally happily coexist alongside them, right? And basically the take home for the conservation is keep your hands off the bats, right? I mean, if you go back to Ebola, for example, the Ebola West African outbreak, the question was, why was it suddenly in West Africa? Well, partly it's in West Africa, probably because we're chopping down all their natural habitat in the forest where they would normally migrate to. So instead of migrating to live in the forest, they go and live in the eaves of houses, and they're defecating Ebola-laden poo in your front door, and bingo, you've got Ebola. So it's not the bats' fault, it's the humans screwing up the habitats for the bats, getting to move to the wrong place. Or in the case of COVID, probably humans bring them into the markets to eat them, right? So yeah, don't blame the bats, keep your hands off the bats and keep their habitats preserved, and then we'll all be happy.
Nothing short of fascinating, that, Tom. You've described your career path. Is it possible for someone to follow the same career path and get where you are today?
That's a good question. I doubt it, for several reasons. First, there's a lot of luck. I mean, It was very unplanned, and a lot of, with all these things, a lot of things just happen by chance, right? So the exact same one, no. But it's, although I didn't really plan it, it's very easy to look back on what I did and see some important things that I would advise people. Firstly, if you want to do a PhD, don't do it unless you want to do the topic. So too many people are out there looking for a PhD because they want a PhD. And to be honest, I mean, I'm talking about biology. I mean, sure, if you want to be some kind of industrial chemist, I'm sure you can do a PhD in anything, and I mean, in chemistry, and go and work there. But if you want to be a pure academic researcher, follow your heart, right? And that doesn't sound easy, because getting money for a PhD itself is a challenge, and some areas are more fun. But there are far too many people applying for every PhD purely because they want to do a PhD, and then they do it, and for them it's like a 9 to 5 job, and their heart's not in it. And that never works out, because this pyramid of academia I was telling about is pretty brutal, and it's always about competition, right? And if you waltz your way through a PhD and don't bother writing any papers and so on, and then it gets to job interview time later for academic jobs or even postdocs who are writing grants, it's a pretty brutal playing field out there. And if somebody did what they wanted to do, was very busy, worked their ass off, has 7 papers, and you've got none, who do you think is going to get the grant, right? So having your heart into it means you're more likely to work on it, because it's your baby. It's no different to writing your novel or something, right? Which means that you will typically have better ideas because you've thought about it more often. I spend a lot of time just thinking, having ideas, right? Because you think about it when you're out running or doing your yoga or whatever. So when you write grants, it's a better story. You've got a better grip of the knowledge of the literature, so you have got ideas that can be tested that are new. You also know who's out there, and people get to know you because of your papers. So that effort you put in, which unfortunately is not a 9 to 5 job, and it's impossible almost to be a real, should we say, at the top of the forefront academic and have a 9 to 5 job. But that definitely makes the playing field a lot less level and puts you on the, should we say, the more likely to succeed part of it, right? And then in addition to the academic part, you have to think about where do you want to work and what does that place need. So there's this idea that universities will just hire the best, they'll hire the smartest of the most, but that's not true. Universities are money-making machines, right? They have to train students. So, I worked out pretty early on that if I wanted to stay in Copenhagen at the University of Copenhagen, I had to work out what do they want. And they don't just want a load of great papers. They want someone who's a team player, who helps run the department, who contributes to all the admin, who does the teaching, all the boring stuff, right? Which has to be done, right? I mean, at a university, our job is to train students. Anybody who's at the university says, my job is just to do research, should not be at a university. I'm completely unsympathetic to that argument, right? Because then they go and work in a private research institute, right? So, of course, my point being, and this is advice I give a lot of people actually, is you can at least make yourself favorable. So, for example, there is a problem actually in academia that people typically have a viewpoint, well, when I get given my permanent job, then yes, I'll start teaching and doing the admin and whatever, right? That's the wrong way to go about doing it, right? It's the way to do it is, while you're covering your own salary off your own grant or whatever, try volunteering and helping and becoming the one everyone likes, because you're not costing stuff and doing stuff, right? Because then who do you think is going to be in the good books when it gets to hiring time? Is it the person who's been waiting to be given their things, or is it the person out there doing the stuff and volunteering, right? And I think this is an extremely important point. People don't like doing it because they're like, well, I get paid so badly anyway, why should I do the extra stuff? But it's again about making the playing field less level and being strategic. And it sounds very, very cynical, but that's the way academia works, right? And so actually, because I run a department, I've actually just finished hiring a lot of faculty, and they're all people that I've been helping nurture get to where they are today, right? But I mean, the people I've held on to, I've had probably, I don't know, like 50 postdocs working with me. And the ones I've held on to are the ones who I can see are the team players, right? It's not just about them, it's about the bigger picture thing, because of course that makes the whole environment better. So my advice is follow your heart, realize the more you do, the better things will be. And don't just think about the academia, think about the collaborations, think about the helping both, other colleagues, but, not just the science, but the other stuff.
Fascinating. A similar question. What does someone need to do to be excellent in your field?
Yeah, well, clearly not being able to recognize plants and animals, because I can't do that. Yeah, it's a very interesting question, because what is excellent, right? I mean, I personally value creativity, right? I think that the really interesting researchers are the people thinking outside the box. Now, the problem is, I actually heard a really interesting talk about this recently, and basically the gist of the talk was pretty much any feature of anything is a normal distribution, right? Whether it's like intelligence or creativity or art skills or music skills, right? There's basically a normal distribution, right? And you've got some people who fall at the extremes because they're really good or really bad. I mean, art, I am at the way crappy, and I'm way off the scale at how bad I am at art, right? So you have this distribution, and if you believe in those things, if you think about creativity, whether it's writing books or doing academia or whatever, it's a distribution, right? Now the problem is, if you are, for example, at the most extreme end of the creativity, there's a very interesting thing that most of your peers are not at that end, which means that when you have ideas, they probably think you're a crackpot because your ideas fall outside what they're normally into. And again, I guess a very good analogy is like avant-garde jazz music or something. I hate jazz music, right? I don't appreciate it, but the real guys on the extreme of the jazz are really appreciated by the guys out there. But most people think that's just a load of crap. I mean, what is this noise, right? And it's the same with creativity. And it may sound silly, but a lot of the time when I talk about ideas, write grants, the people reading my grants are like, that's just crazy. Why would you want that reason? And that's just, you don't need that argument, right? And so, of course, I value the people that really think outside the box, and I typically gravitate towards them. And the guys I work with are this extreme that have these, I would say, very creative, different ideas. But it does put us at odds at the general community, because the general community that's based in the standard theory or whatever is like, I mean, when I write grants on the ideas that we do, I either get fantastic scores or the worst possible score possible, right? Because people say, we know how it works. We've had our ideas for 30 years how it works. Why would you possibly want to stir into trouble and come up with this idea, right? Or they will say like, wow, what a great idea. Let's try it out, right? So anyway, my point is creativity. I think that's super important thing. I'm afraid I've even forgotten what your question was now. I've been so busy talking about normal distributions. But I certainly think creativity is a really important thing, because I think creativity is what may not make the constant progress you need to, tweak and fine tune stuff, which is very, very important. But it's what you need to jump the boundaries to open up the new stuff, right? And ultimately, scientific progress comes with jumping boundaries. The people who really see, for example, the potential of CRISPR, like gene editing, was a big difference to just the regular things going on. And so I think ultimately, I'm not in any way disparaging the people who are not in the creative end, because I am terrible, as I said, at details. I would be the wrong person to tweak and modify and detail, which is super important. But at least I think I can contribute to the occasionally having the crazy ideas to start new areas to look into, and then leave it to much more capable people to actually do them properly.
That's brilliant. Yeah, so Tom, to wind up, do you have anything else you would like to add?
Well, I do actually. I mean, I'm essentially a geneticist, right? I'm a lab person, but I definitely wouldn't be where I was today had I not had the fortune of ending up in the rainforest back when I met you back in Borneo, whenever that was in '97. Because before then, although I'd actually signed up to study biology, I really hadn't done any biology, like real biology, right? And to be completely honest, everything I do today is very much shaped by that. Once you're actually in the wild, in nature, doing proper field biology, you get to see how things work, right? And had I not done that, I would never have had the curiosity or the ideas to start studying those kind of things now, right? So for me, that was massively influential. Had I never, essentially, by my dad being sent to do that, which is another weird story, I would have probably ended up as a regular molecular biologist, just studying, very important stuff, but not what I do now, like how do cell walls work in bacteria, all that kind of stuff, right? So, I cannot highlight enough the importance of biologists getting into nature. And I mean, again, that sounds really corny, but being out there, really experiencing that, even if it's sweaty and hard, and you're like, why should we care about the birds at this canopy, whatever? I mean, it opens up your mind, right? And to me, that was so fundamentally important that I'm extremely grateful I had that opportunity to sweat an awful lot in Danham Valley, pick up a few parasites, and I think I nearly poisoned myself because I had so little money. All I ate for 3 months was rice cooked in river water with soy sauce on it, and I actually came back, and for months I was going to the doctors to kind of get rid of all these salt crystals that accumulated in my kidneys or whatever. But hey, it was worth it.
Tom, thank you very much indeed. I've gone through all my questions. I could make this last on because it's fascinating, but thank you very much.
That's quite right.